Very good morning, students. This is the second lecture in the series on genetics lecture. Okay. Now, this genetics lecture, as I told you in the introductory lecture, last lecture, that they are meant for first MBBS student mainly. Okay. And this will cover only the curriculum of the first MBBS as decided by the Medical Council of India in 2019. So, today's lecture is based on the Mendel's laws of inheritance. Though these Mendel's laws of inheritance are not in that syllabus of MCI, but this lecture is important for understanding of all other lectures which will follow in genetics. Without learning the Mendel's laws of inheritance, okay, it is not possible to understand the genetics. So, though it is not in the syllabus, but I have included this lecture in the series of this genetics just for the benefit of you people. Okay, so I am just going through these laws of inheritance, and for understanding these laws of inheritance, one has to understand the experiments which were carried out by the Mendels, okay, on pea plants. Okay, and I will be very brief, okay, and will not take much time in this explaining this Mendel's laws of inheritance, okay, and then you have to but understand these lectures, okay, very well, and this is given in the book uh, very nicely in the chapter 2. It is very nicely explained, so those who are interested may refer this chapter from the book. Now this is the book, as this is just an acknowledgement of the book. Now coming to the Mendel's laws of inheritance, the Gregor Mendel, who was born in 1822 and died in 1884, was the first to discover the principles by which characters are transmitted from one generation to the next. This was the first scientific study on genetics. Okay, okay. There were so many, I would say, the facts about genetic and transmission of characters were very well known. As in the last lecture, we discussed that the offspring, they resemble each other in their various characters, or they also resemble with their parents. Okay, so many things were already known about the genetic, but the first scientific proof that these characters are transmitted from one generation to next generation, and they follow some rules or some principles, that was told for the first time by the Mendel. Okay, he showed that the transmission of characters follow the statistical law, mathematical rules, and this was quite a bold statement, and nobody was ready to accept this statement that the biological character, how can they follow the mathematical rules or statistical law, okay, as we will see just now. Okay, now, at the time, the details about the chromosome and cell divisions were not known. That means in 18 after 1822, okay, or in that era, nobody was knowing about the chromosomes, about the cell division, okay, and without knowing about the chromosome, without knowing about the cell division, he could determine many characters which are very correct today, okay. He said that the characters are transmitted through factors. Okay, he was first to tell that the characters are transmitted through factors. Factors means what? To which we call today as gene. He was not knowing the word gene, and it was not the word gene was given by him. Okay, he was calling them as factors. Okay, so he said that if a plant is tall, it is because of presence of a factor in that plant. If a plant is short, or if a seed is green, or if a seed is yellow, okay, those all characters are determined by the factors. Okay, now, but he himself was not sure where these factors, to which we now call as gene, are located, because naturally he was not knowing about the chromosomes. Okay, nobody was knowing that within the nucleus there are chromosomes are located, and as we know today that on these chromosomes are sitting the many genes which are responsible for our characters. Okay, right, and this was just not more. Now I'm moving to the next slide. Now, in this slide, I will tell you about the information obtained from the Mendel's experiment. Okay, I have just briefed this information which the Mendel's experiment has to tell us. Okay, so it is very condensed form, but this is quite interesting. Okay, and necessary to understand the genetics. What he said is that each inheritable character is determined by factor. Okay, so two genes he was calling as factor. Okay, now these factors or genes are transmitted through gamete. So he for the first time said that these factors which are responsible for the determination of the characters, they are transmitted from one generation to next, either in the plant kingdom or animal, through the gametes. Okay, that is through the sperms and ovum. Okay, now these factors for each characters comes in pair. That was a quite a revolutionary finding in that era. Okay, when he said that these factors are present in pair. Okay, for a character there are two factors present in an individual plants. Okay, for example, for the height there are two factors. That means character is one, height, and there are two factors for shortness or the tallness. Okay, now then he further said that members of a pair of gene, because they come in pair, the pair of genes he said factors. Okay, the members of pair of genes separates from each other at the time of gametogenesis. So in an individual, these factors are present in pairs and they separate out, okay, from each other when the gametes are forming, okay, in plants or in animal. So each gamete, that is sperm or ovum, okay, or anther, okay, pollen grains, say for example, each gamete contains only one factor. Okay, one factor of a pair. They contain one factor or one gene of a pair. They separate out. Otherwise, an individual, the factors are present in pair, but when gametogenesis takes place, these factors are separated. Okay, so on one gamete will contain only one factor, because you know now that the gametes, they are haploid in number. Haploid means there is the presence of the half of the chromosomes. Okay, so say for example in human being, okay, somatic cell contain 46 chromosome, but gametes means ovum or sperm contain only half number of chromosome, that is 23, and that is the reason the why the factors which were in pair, now they are separated out and they are only one factor present in a in a gamete. Okay, this he has told already with the plants experiment. If plants with contrasting characters are cross-pollinated, I am introducing you a new term, cross-pollination. Cross-pollination means when the plants have two contrasting character. For example, the plant with the short height and the tall height, they are contrasting or cross-pollinated. Cross-pollinated is opposite of the self-pollination. Self-pollination means when the pollen grains from the anther of the same flower falls on the stigma or the ovary of the same flower. That is called as the self-pollination. But in experiment, if the anther, which are the male gametes, okay, of the plant, they are just taken with the help of a brush and put onto the stigma of the ovary or female genital part of a plant, then the fertilization in this way which takes place is called as the cross pollination, and naturally the anthers are taken from one type of the plant and they are put onto the stigma of the other type of the plants, okay, which are in contrasting characters, okay. So this is the cross pollination. I hope you understood what is the cross pollination is that. So if a plant with contrasting characters are cross pollinated, one character is expressed in first generation. So first generation, I will show a diagram. One character is expressed in first generation, and that character is called as dominant in a cross pollination, okay, not in self pollination. And both characters are expressed in second generation. That means dominant as well as recessive, means one which could not express in first generation, both will appear in the second generation when they are allowed to self pollinated. When they are allowed, in the diagram it will be clear, okay, it will be more clear. So thus the transmission of character follows a statistical law. I told you this was a major finding, I mean say, and surprising finding, and people at that time, those who are busy, I mean say those who were scientists and were engaged in the breeding experiments, they were not ready to accept this finding, okay. The transmission of character follows statistical law. That means when the characters, one contrasting character type of the plants are crossed with each other, then the, I mean to say, offspring, they result into 3:1 ratio, okay. That is called, when one contrasting means height, say for height, what are two contrasting character? It is tallness or the shortness, okay. And when these two contrasting characters plants are crossed, they are called as monohybrid, and that means ultimately the offspring which result from this, I mean say, pollination or the cross pollination, that is called as monohybrid. And when the plants with the many contrasting character, or at least two contrasting character, they are crossed with each other, cross pollination is done, then they are called as dihybrid, okay. And then the ratio which will come out, okay, in the offspring, it follows 9:3:3:1, and in monohybrid it is 3:1. 3:1 means, that means in the second generation there will be the 75% will be tall plant and the short plant will be 25%, okay, 20. In this way, for that of the dihybrid cross, the dominant and the recessive character will express in this mathematical ratio. Okay, I will just explain this, okay. See this. This is monohybrid cross, where the only two contrasting characters are there, and the character is of the height. And which are two contrasting? Tall plant and the short plant. And when they are cross-pollinated, let me show you by pointer. When they are cross-pollinated, and I have explained you what is a cross-pollination. That means where you have to make the pollination. That means the anther you bring from the short plant or from tall plant and put it onto the stigma, okay, manually onto the other plant. That is called as cross pollination, so that there is no chance of occurring of the self pollination. You don't allow the self pollination, only the cross pollination is allowed. And how it is prevented self pollination? One has to take out all the anther from a flower, only stigma should remain, and then bring this uh, uh, I mean to say, this pollen grains from the anther of some contrasting plant, and then put it onto the stigma with the help of a brush. It is a quite painstaking experiment which was done by the Mendels for 8 years, okay, for 8 long years he did it, all these, okay. And at that time also the pea plants on which he did it, okay, they were having, I mean to say, only seasonal growth. Every year only one crop is there, okay. So he uh, was to wait for whole year for the next crop to sow, okay. So in this way, this is the monohybrid cross, and tall and the short plants, they are crossed, and this is the cross pollination. And then when in the next year when he sow the seed of these plants, then what result was obtained was quite surprising. That means all 100% plants, they were tall, all 100%. That means this character of tallness was dominant over the character of shortness, which could not express, and this character was called as recessive character. Okay. Now then these all tall plants flowers were then allowed to grow, and then they were allowed to self pollinate. Okay, self. Okay. And when this in the third year, when this again the plant were, I mean say the seeds were sown, the second generation result were obtained. Okay. Now, and they were quite surprisingly different than the result of the first generation. Okay. And these were that 75% were tall and 25% were short. That means this character which was recessive, which could not appear in first generation, all of a sudden appeared in the second generation without any alteration. That means it has not taken the some tallness or some I mean to say it has not shown medium growth. Okay, it was as short as it was in the first, I mean say pollination, cross pollination. So characters were not blending. Okay, they were not I mean to say inhibited. They were uh, fully expressed, the full tall plants and full short plants. Okay. Now, if you pollinate them in the fourth generation, self-pollinate again, then again the ratio will remain the same. Okay, if you separately sow the seeds of this salt plant, all 100% will be salt. Okay, because there is no factor for tallness as we will see just now was present. Now, when you will sow the tall plant, out of this 25% will give 100% tall, but remaining 50% will give the same ratio which was obtained here in the second generation. Okay. Uh, this this ratio is then again 75% tall and 25% dwarf, and again 75% tall and 20. If you just do the calculations, okay, of these plants, ultimately the ratio will come is 3 is to 1. 75% tall and one I'm say 25% dwarf. So this is exactly following the mathematical or statistical laws. Okay, and Mendel could predict that why they are following these mathematical laws. That's why he said that these factors which determine the character, they come in pair in an individual. In an individual they come in pair, and they separate out when the gametes are formed. They separate out when, and on the basis of this he could just say that yes, it is possible, and this will follow the mathematical laws. Now, when we come to very shortly, I'm just very telling you very superficially and quickly will pass. The dihybrid cross, okay, where I told you dihybrid cross means when the pollination is met or cross pollination is met between the two contrasting character. For example, here is the pea plant with the pure bred yellow round seeds. Yellow is one character, okay, of the seed color is one character, and its roundness is another character. Both characters are present in one seed. Okay, both characters are there in a plant. Say for example, they are present in a seed of a plant, and when they were cross pollinated, okay, with all those experiments, they were cross pollinated with the plants which were having the seed which was green in color, and its surface of the seed was not smooth but it was wrinkled. Okay, so these are also two characters. Uh, and when these two contrasting character plants, they were cross pollinated, then in the first generation, what was the result in the seed? Which type of the seeds they were obtained? They were all pure, all hybrid type of the seed with the yellow color and the roundness, that means a smooth surface, round surface, which was exactly like this. Okay. Uh, okay, yellow and the round. So this character of the yellowness and the character of the roundness is a dominant character over what? Over the green color of the seed as well as the wrinkling of the seed. Okay. And when this again were sown, these all 100% seeds which were yellow and the round, they were sown again, and in the next generation or in next season, when the seeds were collected, these seeds were have shown different characters, and these I mean say plants were self-pollinated. Okay, they were not crossed. In the for second generation, they were self-pollinated, and when F2 generation, that means second generation, where seeds were collected, and when he started counting them, then he got 9 ratios of the yellow and the round seed, then 3 ratio of the wrinkled and yellow. That means here now two characters have come in one seed. Okay, which were the different characters. Here wrinkleness has been obtained from this seed here, and then it is color was obtained from this. So the two characters have come close to each other, right? This is very important. And then again a combination of character has obtained in 3 ratios of these seeds. That means smoothness, it has come from this one which was yellow in color, but this color has come from the seed which was wrinkled also. Okay, wrinkled. And then in one ratio, one obtained both the recessive character which was the green as well as wrinkled. Okay, green and as well as wrinkled. So what is the ratio of this seed in the second generation? 9 3 3 1. 9 3 3 1 ratio, and this were the result again which were constantly coming again and again in the dihybrid cross, and these are also following the statistical laws. Okay, they are statistics. Another important thing which is said by this experiment is that the characters they don't blend each other, they always remain in their pure form. Wrinkleness will always remain as wrinkleness, even if it is transmitting or coming in combination with yellow or the green color. So is the shape will always, I mean say color will always remain constant. It will be green in the green seeds and yellow, pure yellow and pure green. It will never happen that some yellowness and some greenness will mix together and a new color will form. Never, never. So this was in very short, this is a dihybrid cross. Moving to the next slide. Now, these are the laws of the Mendel's inheritance. Okay, the Mendel himself has not given these laws. Okay, these were given later on in 1900 century when the Mendel's experiment findings were rediscovered. The poor fellow gave just a talk in front of around 40 botanists who are sitting in one fine evening, and then everybody heard it and nobody believed it. Okay, and then Mendel's also forgot his own experiments, okay, after its publication in a small journal of Europe, and then everything was forgotten. But all of a sudden in 1900, many peoples they rediscovered these findings again without knowing that these findings were already there, okay, by Mendel's. But when they search it, they got it. Oh, Mendel's had already done it, everything, and their finding was also similar to what Mendel's has observed 35 years back, because they just published their paper. Three different peoples from three different countries of Europe published the same findings. Okay, and they acknowledge, of course, to that in review of literature to the Mendel's. Mendel's had also observed the same thing, and that's why these laws, I mean say his findings, okay, then were presented as the laws of inheritance, okay, Mendel's. Okay, and this gives the recognition to Mendel and his experiment after he died. Okay, after, and that's why now the Mendel is called as the father of genetics, modern genetics rather. Okay. Now these are the three laws, and I will try to explain you, though it is not in your syllabus. The laws of uniformity. Okay, I will just read it out. When plants with two contrasting, say shortness and the tallness, okay, shortness and tallness are crossed. Okay, cross means cross mated. Okay, or cross pollinated. They are, then these characters do not blend. That means there is no intermediate height. The first generation were all tall, and there was no of the intermediate. And when the second generation, the seeds were sown, okay, 75% were tall and the 25% were short. So there was no uh, blending of the character has taken place. Okay, and if any character does not express in the first generation, as we have seen in the first generation, all 100% were tall, and it was called as a dominant character over the shortness. It may reappear without change in subsequent generation, as we have seen in the second and third generation. The short plants, they reappeared. Okay, they reappeared. Now, the second law is the law of segregation, which Mendel has said, okay, very clearly. When genes of a pair are separate, he said that the factors, they come in pair, and two factors we call now as gene comes in pair, and they separate out at the time of gametogenesis, and thus only one member of the pair goes in a sperm or ovum, or mm, into the gametes. Okay, into the unaltered. Okay, there's completely unaltered. Okay, there is no change in the characters. Okay, in the gene characters. um, Now the last law is the law of independent assortment, and this law is based on the dihybrid cross, which we have seen in the yellow round seed and the green wrinkle seed. On that basis he could form this third law. Okay, at the time of gametogenesis, members of different genes pair. Now he is not calling a single gene pair. He is calling different genes pair assort independently of one another. So the greenness has assorted. That means always the greenness was not associated with the wrinkleness, or the yellow color was not always associated with that of the roundness, okay, of the shape of the seed. Now in the second generation, we saw them to be appearing in the ratio of 9:3:3:1. That means 3:3 means where the characters uh, were just coming from two different sources. That means the wrinkleness and the yellowness, which was not in their original plants, and the greenness and the smoothness was there. Okay, so they are, these characters were independently, they were not always fixed. Oh, we will not go separately, we will go together, that they were not saying. So the characters were going independently. Okay, so this is called as the law of independent assortment. Just I will take few more minutes to explain. Hmm? Now what happened after Mendel's? Okay, because this experiment was, I mean say, I told you, was published in 1865. Mendel gave his finding in 1865 in a group, and then it was published later also in the same year. Findings of his work was unappreciated till 1900. I told you why it was unappreciated. We have to see that. Okay, but when the three other peoples, they did this experiment and got the same result, then this was realized. Okay, and now the big question is that why these findings were not appreciated in 1865 when he published these findings. Okay, of 8 years of hard work uh, had gone waste. Okay, now the reasons are many. Okay, the first reason is that importance of nucleus in cell division was not known. The cell division was not understood. Okay, that nucleus contains the chromosome, it was not known. Okay, it was only suspected, it was not known for sure, and when Mendel was doing his experiment. Okay, so importance of nucleus in cell division was not known. If it would have been, there would have been no problem in understanding his findings. The chromosomes were not discovered. At that time, nobody was knowing what is chromosome and where they are located. Where they are located, nobody was knowing. Okay. Details about the cell divisions were not known. The faint idea about cell division, because there were not a good quality of microscope also at that time. Okay, so details about cell divisions were not known. Nothing was known about the reduction in chromosome during meiosis. Nobody was knowing that there is some kind of a cell division called as meiosis, where the reduction in number of chromosome takes place in the gametes. Okay, when gametes are formed, they contain half number of chromosome, was not known. If this would have been known, Mendel was enough intelligent to tell this fact, that why there occurs the, I mean say, why a pair of factors separates at gametogenesis, and then only gamete contain only one factor. Okay, this he could have been very well correlated, which he has thought without knowing anything. He has thought of it. Okay, it was also not known that the genes are present on chromosomes. So nothing was known. Okay, so just by experiment he deduced, he could imagine that this must be happening. Okay, this must be responsible for the result which I got from this my pea plants. Okay, so let's come to the next slide. Probably it may be the last slide. No. After the discovery of Mendel's in 1965, in 1873, uh, essentials after the discovery, these things were found. Okay, in 1873, essentials of mitosis was discovered. Okay, so cell division was discovered after the Mendel's experiments. Okay, in 1879, it was discovered, and when the Mendel has presented his finding, 1865. Okay, it was discovered that chromosome splits longitudinally and migrates to daughter cells during the cell division. Okay, in 1883, it was discovered that the somatic cell contained diploid and gametes contain haploid number of chromosomes. Okay, so this was quite late after the discovery of Mendel's, when one. So when these three scientists who did this experiment in 1900, they were knowing all these things. Okay, and then they did this experiment. So for them it was very easy to just understand the result of their experiment, which was not easy for Mendel's. Okay, to understand this result. Thus by 1900, scientists were ready to discover, okay, where genes are located. Now they are in this position. In 1900, they were in this position to discover where these factors which are determining this character are located in the cell. Okay, they were still not knowing, but they were now ready to discover it. Okay, they were ready to discover that means that much knowledge was there, so that started discovering, oh, where are genes. Okay, what determines the factors. I mean say the characters are factors, but these factors are where. Okay, and then this is the last slide. Where are these factors located? This was the big question in 1900. Okay, and people started thinking with their background knowledge of cell division and reduction in the number of chromosome in the gamete formation. Okay, their mind was now fertile, and they started speculating. Okay, genes must be here or there. Okay, then there came a medical student, okay, in 1902, who gave the chromosome theory of heredity. Okay, and he was a medical student. Later he died of appendicitis. Okay, and he became, after passing the, I mean to say, medical, a surgeon and then died of appendicitis himself. Okay. Now he was a Walter S. Sutton. Okay. There was some other fellow also during this time who also gave this, what he also said the same thing which this Sutton has said. Okay. The factors to which we call as genes are physically located on chromosome. So the fellow said that the genes are physically located on chromosome. He was the first person to say. And why he could say all these? He could say all this because at that time, okay, he was knowing about the meiosis. Okay, he was. And why he said so? Reduction in number of chromosome haploid at the time of gametogenesis and restoration of diploid number at the time of fertilization explains the Mendel's observation. Okay, so now he has also not seen the genes, but he has speculated, and it was just a hypothesis that said that chromosome theory of Herod. That means chromosomes are responsible for the hereditary or the transmission of characters from one generation to next, and these chromosomes are transmitted from one generation to next. That means you have inherited the sperm from your father and ovum from your mother along with their chromosomes. Okay, along with. So this completes the lecture today. Okay, and I hope that you have understood. I have put you onto that platform now. Now in the next lecture, since he has said that chromosomes are the, I mean to say, responsible for our heredity, now we will just focus on chromosome in next lecture. Thank you very much.